Students often get mixed up here. This mini lecture is on the proteins that are embedded in cell membranes. So the functions listed here apply only to proteins embedded in cell membranes, not all proteins. And this is why you see the gray phospholipid bilayer in each of these pictures. And on the bottom right are even labels. It labels the extracellular matrix on the outside of the cell. We will encounter all types of proteins embedded in cell membranes. The most often encountered is the first listed here, channels. Pathways for molecules to cross the phospholipid bilayer barrier. The rest of this mini lecture goes into much more detail about the various types of channels that we will encounter in this class. But before we list those, let's just be sure to take another look at these functions, which are functions of cell membrane proteins. These are not functions of all types of proteins. We are familiar with enzymatic proteins from a previous mini lecture. We know about enzymes that might float in the blood, but these enzymes instead are embedded into the cell membrane instead of free floating around the cell or in the blood. They usually have some part of them, some structure dangling off into the cytosol where they catalyze their reaction. When we get to speaking about hormones and the immune system, we will talk about receptor proteins that are embedded in cell membranes and respond to chemical signals. For example, your heart makes a hormone. No, actually, let's strike that. Your brain, it is your brain. Final answer. Your brain makes a hormone called the antidiuretic hormone, and it helps you conserve water when you're dehydrated. This hormone, it floats around your body, but it only communicates with cells in your kidney. No other cells, only kidney cells, have the specific receptors in their cell membranes that would help them receive this signal. Your immune system has cells that are capable of recognizing your own cells. Your cells have specific proteins embedded in the cell membranes, and those proteins are almost like a sticker that says, "Hello, my name is Amy." You know those blue stickers? Ruined many a sweater with those. In many autoimmune diseases, your immune system stops recognizing the identity proteins on your cells, or the identity proteins on your cells begin to be made in a malformed structure. Either way, your immune system is no longer able to say, "Oh, that cell is made by Amy," or whatever your name is. When we look at the heart, we will see cells that are bonded together and contract in a coordinated fashion. These cells have structures that are called gap junctions, and they allow these cells to share cytoplasm. And the last type of protein that we find embedded in cell membranes are these anchoring proteins. These proteins are capable of connecting or anchoring the cells to things in or outside the cell. So it can bind to the cell's cytoskeleton that's on the inside, or as the picture indicates here, some collagen fibers that are actually on the outside of the cell. All your cells have protein channels embedded in their membranes. The various protein channels, they contain a type of protein channel called a leaky channel because it's always open, right? Like a leaky sink. That reminds me something about my kitchen sink. Anyway, channels are specific to the size and shape of one molecule. So this pink protein here embedded in the cell membrane only allows one type of molecule to move through it. Other molecules with different shapes, different sizes, different charges, they have their own protein that allows them to move from one side to the other. Which direction? Like, is this a one-way street or a two-way street? Well, which direction the molecule moves in is determined by the gradient, which we talk about in another mini lecture. But you have these leaky channels in all of your cells, and there are channels that allow sodium to leak into the cell, and there are channels that also allow potassium to leak out of the cell. There's also protein channels that allow calcium to move according to its concentration gradient. There are channels that will only open when a certain molecule connects with it. I have a picture of a cell membrane here. We'll just label the outside and the inside of the cell. On the left is a channel that is closed. So despite there being an appropriate gradient, a chosen molecule would not be able to move through this closed protein channel. On the right, I have an open channel. And of course, the chosen molecule can move right through this open channel according to its gradient. But notice that there's a molecule that acts as a key, and it's opening the channel. That molecule is different than the molecule, ion, or atom that moves through the channel. So the shape of the protein channel determines what molecule moves through it. But the shape of this active site, which is a term that came at us in chapter 2 about enzymes, the shape of this active site determines which molecule will act as a key, opening up the protein channel. These types of protein channels are called chemically gated channel. The term chemical here just indicates molecule or item or even thing. It depends on what book I'm teaching from, but sometimes they can also be called ligand-gated channels. I find that kind of funny because ligand's like this old term from chemistry, and basically it just means thing. I just feel like there were some people sitting around a table and they were like, I don't know what to call this. And someone said, ligand. It's a word that means nothing. Whereas the previous channels would open with a physical molecule making a connection, like a key that's inserted into a lock, these types of channel proteins only open due to a change in voltage. And we'll just label, here's the intracellular fluid, here's the extracellular fluid. We'll put some sodium ions outside because we know they're abundant. Let's put some potassium on the inside because we know that they're abundant as well. These little voltage-gated channels, you may not realize this, but this is how key cards work at hotels and other places. There's actually an active magnetic field running through the lock, and when you insert the key card, you break that field, opening the door. You are literally opening the door and walking through it by using a change in voltage. As far as cells are concerned, voltage or electricity, it's just the movement of ions. And we know that sodium ions and potassium ions are leaking into and out of a protein, out of a cell all day, right? And these are positively charged, so they're changing the voltage. These channels here, they have something like a threshold of voltage or a set point of voltage at which they will open. So if enough sodium ions leak into the cell or enough potassium ions leak out of a cell, then these voltage-gated channel proteins will open, allowing their specific ion to flow according to the gradient. So these voltage-gated proteins, they work much in the same way as the chemically gated proteins. They open or close upon demand, and they allow only certain ions to flow. But the difference is what triggers them to open either being a physical molecule that sets into an active site or a voltage threshold. We have greatly simplified things in our examples, but in reality, transport is much more complicated. For example, the management of glucose by the kidney is linked to the management of sodium. Co-transport is like when somebody hitches a ride with someone else. And you can see a couple examples of co-transport in the examples on the left with the blue membrane proteins. 2 or sometimes even 3 molecules are being moved at the same time. On the right side of the picture is something called antiport. And this is when one molecule or ion moves in one direction and another moves in the opposite. There is this little protein called the sodium-potassium pump, which we will constantly be reminded of in this class. And this pump is an antiporter, transporting sodium back to the outside of the cell and potassium back to the inside of the cell. And it's worthwhile to mention that co-transport and anti-transport, it can be active using ATP or it can be passive.